YouTubers, hello, YouTubers and students. Dave Soriano. I'm a chemistry professor with the University of Pittsburgh's Bradford campus in Western Pennsylvania, USA, and I do molecular modeling. And because of the social isolation and my need to teach via remote education, I decided that there are aspects of the molecular modeling that I would like to share with you to generate ideas and for fundamental instruction for undergrads. I use Cygress platform through the University of Pittsburgh molecular modeling platform. What caught my interest at the Protein Databank, which can be located with your browser, PDB, Protein Databank. I've been using it now for, I don't know, probably since it had 8,000 entries. It's up to 163,000 plus now. X-rays, NMRs of proteins and other materials. So I found catalog number 6VSB, and this was uploaded by Professor McLellan at the University of Texas Austin and co-workers at the NIH, and this was uploaded in February 10th of this year, 2020. The paper can be found in Science, volume 367, published this year, 2020, pages 1260 to 1263 in this landmark paper. And it focuses, and I'll use very simple undergraduate terms, it focuses on the so-called spike protein, which is on the entire surface of the coronavirus. And the spike protein is a glycoprotein, and it's comprised, actually, it's almost like a rocket. It's comprised of S1 and S2 domains. It's a trimer, so it's 3 units of the same protein bundled together. And Professor McLellan and his group, they were able to upload a view of the pre-fusion before docking to the ACE2. From our perspective, the human cell, ACE2, angiotensin converting enzyme 2, which is an enzyme embedded in the cellular membrane. And this is pre-fusion, so you're not going to see the ACE2 receptor site in this image, but it's the 2019 N-CoV spike glycoprotein with a single receptor binding domain up. Now, in their paper, you'll find that when docking, considerable conformational changes take place, and the spike protein, glycoprotein, is almost like a rocket. There's the S1 and then the S2 portions. And what I want to just show you in this simple exercise, just to generate ideas, a cursory view. I downloaded the file, the PDB file, and now we'll proceed to my Cygress, which I have open. And I am going to open this up. And quite arbitrarily, I'll go to, an open file. There we are. And I'm going to import that file. So there it is, the 6VSB PDB. Let that open. And there we have it. Now, it's important not to get intimidated by this. And if you're doing computer-aided drug design undergrads, what you learn in this very exciting field of computer-aided drug design, you would have a ligand, let's say an inhibitor or a natural substrate embedded in the active site of an enzyme. Then you would cut out all irrelevant portions of the protein and zero in on the active site and the ligand and begin to do docking and statistical fits. Now, we're not going to do that here. What we're doing in this exercise is simply taking this. I'm going to convert it to spacefill. There's many, many things you can do. And right now we're going to go to simply space filling. And there you have the image uploaded by McLellan and co-workers. And I just want to show you the surfaces here that are accessible surfaces on this protein. We're using a rollover probe of water, 1.4 angstroms, a probe radius that can be used in the calculation of the accessible surfaces. Connolly was a theoretical person many years ago, the Connolly who first started to use and develop programs to do such a thing. This can be used for remote inhibitors in rational drug design, where you have a non-competitive candidate drug that binds on the surface of a protein, inducing conformational changes, preventing the substrate from accessing the active site. But that's another story at another time. So let's just see where we get these accessible surfaces. And they're portrayed in this pinkish red color. What I'm going to do is simply rotate it around so you get a feel for what you're seeing in terms of surface accessibility. So we'll rotate this around, and you can begin to see what the possibilities are. Almost like a flyby before we went to the moon. You fly by craters before attempting a landing. Now, Skidmore in the United Kingdom, Professor Skidmore published a paper where he found pockets of positively charged residues, arginine, lysine, and in fact, they were in the 300 to 400 series of the sequence. He found a pocket of a physiological pH 7.2, a pocket of positively charged residues that could serve as what he called a patch, where heparin, members of the heparin family, could bind to this spike protein and enable it, disable it. But that's another story. I'll do that at another time. We'll review his work. But now I just want to rotate this. And of course, you can also access with Sigras amino acids that are there. And you can point, there's aspartate 420 right there. See the lower right of my desktop? And you can get an idea of what's not accessible and what is. So there's alanine 435, and that's an accessible crevice. And these crevices could be of great interest in rationalizing some candidate that could bind to the spike protein and prevent its conformational changes when docking to ACE2. So that's where I'm at with this, and I'll be doing a lot of computations here, and but you can see there are a lot of crevices. You're looking at the chimer, and of course you can keep the flap open, the one that's ready to dock, and delete all other portions of this if you simply wanted to study the amino acid sequences and information around the critical docking portion before the viral RNA through S2 is injected into the host cell. First, the membranes merge. But this might give you some idea, and there may be some scientists, researchers watching who it would generate ideas, and certainly researchers that would know a lot more than I would. I've been modeling for about 20 years, aldose reductase enzyme in particular. But that would give you an idea of what you can do as an initial start of thinking how we could induce conformational changes in this spike protein before it docks to a human ACE2. Any comments or questions, you get a hold of me and let me know. Thank you so much for watching. I'll upload more videos of this nature. Bye for now.